moving a bit to biology. Then apparently she had a very important trip to the Amazon, right? You report on your webpage, so I guess it's very important.
I have to check my webpage now.
And after that, she moved to the John Innes Center in Norwich, where she opened her own lab that was leading, and this lab was including an interdisciplinary team involving physicists, mathematicians, biologists, so not so easy to combine everything, I guess. And then in 2018, she moved to Cardiff, where she's now a reader. Now, I understand that she's an aficionado of adaptive complex systems, so she worked on that. And her goal, let's say long-term goal, is to understand and unravel the origin and impact of cell and tissue polarity in developing organisms, right?
Really good.
So today's talk is probably about that. So from cell polarity to tissue polarity.
Thank you very much, Dario. I think you're better than ChatGPT and like, whoa. Barely recognize myself there. Okay, so today we're going to talk about cell polarity going to tissue polarity. So this is a theme that tomorrow for the students will go a little bit more calmly through the cell polarity part. But what I want to do with you guys, so you have to sort of bear with me on this journey. So we're going to talk about mechanisms of cell polarity in different species and start looking at analogies. Then we will think about what happens when we put cells together, and we're going to look at different situations. And once we accept the fact that we have tissue polarity and that it's very ubiquitous for development, we're going to start looking at a case study where we look at the constraints that you get by just being polarized as a tissue. So I like to just put up the names of the students that have been working on various parts of the story over the past. And these are my collaborators. So long-standing one is Stan Murray, Leah Cachette, Alan Champneys, Rico Cohen, and Andrew Bangham, who no longer with us, but he did a very important part in some of the frameworks I'm going to show you, and Toru Fujiwara in Tokyo. So you guys are sick of my little cartoon here, but again, just introduce me to those that don't know me. I'm very interested in understanding morphogenesis, so the creation of complexity and form and specialized tissues coming from something simple, which is just the information in the DNA and a little bit more. So how does that complexity unfold? And to do that, because there's so many nonlinear and non-hierarchical feedbacks, we really have to use a mathematical approach, and because we want to understand shape and feedbacks between shape and biochemical patterns, we need to use physics. So basically, to do this little miracle here, cells have to divide, they have to become more, and that means that they have to know when to divide and when to stop, really important. So this is like biology 100. They have to move to different places, so we have to think about chemotaxis. How do they know how to move to places, and how are they aware of that? They have to differentiate into the appropriate cell type, so that's a whole different part of our research that I'm going to talk about, which is stem cell differentiation and decisions that they make on their gene level. They have to then also, as a cohort, expand in different directions, so there's a lot of growth and mechanics there, which we have to understand for the proper shape coming up. But what you also have to realize when we think about development, the whole time this thing has to function. Yeah? So there is also not just the shape which is emerging, but there is a reason why those shapes are there, and there's also physiology. And development and physiology and function, they're usually in biology two very separate fields. It's like, I don't know, people working on soft matter physics or quantum mechanics, you know, and sometimes, you know, we have to do some connections there, and that in biology is a big thing by itself. Okay, so to do all of this above, what I want to tell you is that cells will need to communicate. Yeah, they can't just obey their little instructions that they have inside them. They're actually the whole time aware of the environment, and they're communicating with one another. They're chit-chatting a lot. Yeah. So this is something we want to understand. What we also start realizing is messages they move around are sometimes very dynamical and sometimes very directed. They have spatial patterns. Other thing that you saw this morning, they have to stick with one another, right? So they have to become coherent. And for all of these things to happen properly, they need to have polarity. Okay, so what is cell polarity? So if we think about the cell as the basic building block of life, at least organisms, we see that there's a difference here is because these Lego building blocks, they already come with a certain type of break of symmetry, right? We already take that for granted, and that's why we can so easily staple them on top of each other. But so a cell, which for all purposes in this talk, we're just going to think about it as a, this membrane, this bag of cytosol, right, which doesn't really have a structure in it yet. Yeah, it needs to polarize too, so it needs to have the sense of direction, to move directly, to do its functions, its communication directly. So when I talk about polarity, working definition, I mean that we're going to have an asymmetrical distribution of some chemical species, probably a protein. Okay, and we want to understand how those, because everything is diffusing, by itself homogeneously usually. And cells, they can have a rest state, they can be apolar, and then they can polarize. So we know that these things also happen. So the question is, how can we understand how this polarity comes forth and the different properties it has? So this story began when we were looking a long time ago, actually. I was not working on my PhD, but talking to other people on a subject that had nothing to do with my PhD. But so Alexander Verkovsky, and he's a biophysicist who looks at the biophysics of these moving cells. These are single cells of the immune system of the fish, of all things. But in fact, we also have them. So if you have a wound on your skin, these cells will migrate to the wound. They perceive the wound is there, and they go, yeah? And what you have to know about these cells is they start up looking like a fried egg, where nothing is happening. They have sort of like a little bit of a nucleus here. And then, when they know they have to polarize, off they go, right? So they'll have a back, and they have this leading edge, right? And if I would make a cross-section of this cell like this, you'd see it has like its nucleus here, and it has this very sort of long lamellipodial structure that goes forward. Okay, so it has this beautiful direction, it has quite a nice velocity, and it can go great distances to go to the wound site and eat up all the bad bacteria and stuff which are coming in. So one thing you have to know, though, in this experiment, which is really interesting, Sasha Verkovsky, what he did, he took away the nucleus. Yeah. And it still works perfectly well. Okay. So why was it so important for us? Because you don't need to worry about remember what I told you guys the other day, the transcription, the translation, right? We just have the proteins in that cell doing its stuff. And it can perceive gradients, electric gradients, and it moves in this characteristic way. So in the end of the day, what is making it go forward? So we have cytoskeleton at the leading edge. These little rods, they're being additioned the whole time. So you have filamentous actin, and the monomeric actin, right, gets stuck to the front, so it grows. The membrane is fluctuating, and like a ratchet, so it's quite a thermodynamic process. Each time that the membrane is a little bit further apart, there's some space for that to grow, and then it can't move backwards, right? So you have this sort of process happening there. And so you have these little sticks poking forward, and you get more and more of them because they like to branch. So something can bind here, and they branch with a certain angle. Those things are called ARP23, so those are the things which allow those little poles to branch forward. So by promoting the branching in the forward and allowing this to grow, you can have this protrusive force. But now you guys will ask, aha, but why don't we have it growing backwards? Yeah. So backwards, for some reason, these little filaments, they're being capped. Yeah. So so they're not growing that much, so they're not able to grow, so they're capped. And also, we don't have the abundance of those ARP23s, those things. So we have much fewer pointed ends. We get a selection of them to the front. But then you can ask, why do you cap them in the back? Who's doing that, right? Who's telling the cell to cap them in the back and let them grow in the front? Okay, so we know then that there are these small g proteins, and there you have to go back to the things that Erwin has been telling you guys about the MINS system in the bacteria. So these, I'll go a little bit into them, but so they're going to be switches. So these are basically proteins that they can have, right, an on state where they're GTP bond, and then they can do things. So this will be the membrane. This is your little protein. Yeah. Then it can have downstream processes that it activates. In this case, for example, CDC42 and RAC, they do things like promoting ARP23 to be there. They also inhibit the capping happening. So you can already see that these are really the guys you want to have in the front, because they do things which a front should do. And this here, this row, he is the one which is actually also allowing your cytoskeleton to contract through some motors. So that's doing contraction, because this is not only moving to the forward, it's also retracting in the back, okay? So I'm sort of compressing a lot of biophysics here, right? But then the question that you have to ask is more like, okay, so if we have these proteins, they can be in the membrane, active, and they can diffuse around in the cytosol. How do they position themselves? What is positioning themselves? How does that pattern emerge? Okay, so what are these arrows here? Now, for every cell type you're going to see, you're going to find small g proteins which are linked to polarity, which are doing these things. But they might come in with different names. They might be a little bit homologous, a little bit different to those. And they might also have different interactions, because these arrows, this is their crosstalk, right? So when we have a positive arrow here, like just an arrow with an arrowhead, we mean that CDC42, this guy here, he activates this, and this here activates that guy. And now here we have these cross inhibitions. Okay. So what do I mean with this? I'm just going to go quickly through it. So as I was telling you, this class of proteins, which are everywhere in bacteria, in plants, in humans, in any cell we know, they will have a active state where they do their effectors. They can be inactivated, so this is why we call them a switch. So with this guanine, they can be activated by the guanine exchange factor, and then they can be inactivated here. And once they're inactive, they still linger a little bit in the membrane, and then someone can come and take it back into the cytosol. Okay, that's called the GDI. So I really don't want to bore you guys with biology, but just to say, this is sort of the background. We have a network. Don't worry about the arrows. Just realize they're going on and off through these gaps and gaps, and we have the crosstalk. This is coming from the literature, but it's, doesn't tell us much. So what we do is we put them, as you guys have seen for the MIN system, you write them down in reaction diffusion equations. So what are we seeing here? First of all, I'm going to use also that, so this was work that I did during my PhD, so 2007. So what we do here is we realize that these small g proteins, they're going to have a form which is active and a form which is inactive. But because Verkovsky took away the nuclei here, we know nothing's being produced. Right? On the time scale of that movement, we'll just say the proteins are stable, they're not being broken down. There's a conservation of the amount of these proteins. So we can only so if we describe here the active forms, right, we know immediately what's going to happen with the inactive forms, because active plus inactive equals the total of that population. And please, if anybody has any question, really just put up the hand, yeah? So here we have the equation for CDC42, for RAC, and for Rho, for the three species we have there. Right? And so these would be the diffusion coefficients of them on the membrane, right? DM, 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 all the same. This term here is telling you how they're becoming deactivated. So this is CDC42 becoming deactivated, RAC and Rho. And these terms here show how the inactive form is becoming activated. Okay? So what we see here is that in the case of CDC42, it's being repressed by Rho. Right? And it has this parameter here, which is a basal activation rate of CDC42. In the case of RAC here, we see that CDC42 is activating it, in fact. And the case of Rho here, we see that, it's being activated by RAC, but it's being inactivated by CDC42. Okay, we try to put quite parsimonious mass action, kinetic terms, but I put a little n here, and you can say, why did you put this Hill coefficient, right? Why did you put this Hill coefficient there? You don't know if there's cooperativity, you don't know how many molecules are coming together. I did it because in the very beginning of this work, we thought that to have a polarity, we would want to have the system give us some type of bistability. Okay, how naive. We thought that we needed to have a state, a front state, in which CDC42 would be, if we just look at the reaction part of that equation, the well-mixed case, would have a stable state high, and then we would have to have another region where it would have a stable state low. Yeah? So if we look at, for example, CDC42, the equilibrium value of it, and we're looking at a bifurcation graph where we're changing this basal rate of activation, what we see, it gradually goes up with a Hill coefficient of 3. But we could get this very nice sort of fold here right? So we have this bistability for a bigger Hill coefficient. So we have a lower branch here, right, for a certain amount, for a certain parameter here, where you would be at a, you know, at a stable low amount of CDC42, like the back of the cell, and a high one, like the front of the cell. But obviously, what this is telling us is just that some cells are going to be high and some cells are going to be low. It's not telling us that we're going to have these phase separation happening in the cell. And in fact, when I was making these plots back then, so this is a chemograph, so this is a time-space plot, so this is space and this is time. What we see is even if we would start it with a little, like in this case, where we know that you have this bistability, if you would start here the high phase and the low phase, what you see is obviously you have this traveling front. The high thing takes over. So this cell becomes all up, right? And here it doesn't start. So we were just thinking, okay, can we find a really fast way to look at these networks and to ask the question, would it possibly be able to give me that pattern that I want? Working definition of pattern. I have a front and a back. Okay, this is it. So we wanted to have something where you can sort of probe if it would be able to give you this, yeah? So what I did then is I used a thing called local perturbation analysis, but it's not they're different perturbation analysis, obviously, but this is where, okay, I'll explain what I mean here. So we have the active amount. Imagine here, this is the spatial cross-section of your cell. You know, everything is well mixed. We have a homogeneous equilibrium, so we have this active CDC42 amount here, which is slowly diffusing because it's in the membrane. You have your inactive CDC42, which we know is quickly diffusing because it's in the cytosol. As Avrin said, maybe 100-fold more. And now we imagine what happens if you would make a perturbation at a patch of that cell, right? So you have a lot more in a certain place. And it's arbitrarily small, but it could be of a finite height. It could be actually quite large. It doesn't have to be a small perturbation. Okay, so we're activating this little patch here. That means it's taking it away from the inactive pool. Yeah? Because the sum of this here has to equal total, right? However, now already start saying that I'm talking about what's happening with the concentration locally, what's happening with the concentration globally, both for the active and for the inactive form. But remember that because the inactive form is so fast, for a moment I can just go to the limit where it's infinitely fast. So in that case, I'll just say that the concentration here locally equals that concentration globally, right? Because it smoothens out, yeah? That whole thing will go down a little bit, but we have quite a big cell, so we don't even have to worry about that too much. So that's sort of the approximation we do. Okay, so if that's the case, I can start now writing down equations for a local patch and for a global patch, okay? So these will be two different variables now, but they both look at the same CI, because CI global is equal to CI local. Okay, so if we do this little exercise, so we had before our equations, the global ones. We copy-pasted, we just call it local, local, local. But now the global species, they will be looking at the global levels of the other small g proteins, G, G, G. The local only looks at the local levels, L, L, L. But both of them are coupled together because both of them are looking here at the inactive forms, which are the same everywhere. So this is the global level. This is at the perturbation. And so this is what I just want to say. So this will be the same. And then suddenly our bifurcation becomes very different because now we have the black is our homogeneous state. And this is what's going to happen to the patch. Okay? So what's really nice now is that you can see, for example, if I have this parameter regime here, a low amount of activation, I'm never going to perturb my cell. My cell will always be in a rest state. If I come in here, it is possible, okay, to trigger it to polarity, but it won't happen spontaneously. Yeah? In this regime here, I will have spontaneous polarization, right, where you have a high amount and a low amount. And if I continue here, right, either you're in this branch or you're going to, again, continue in a global. So it was really nice, if you go back here to our system, we see that, okay, for n equals 6 now, we actually get our front and our back, and we get this effect which later, talking to Leah Cachette, and she had a postdoc which was working on it, he termed it as wave pinning. Okay, this is wave pinning. But what was really nice So wave pinning is this, because our high-level patch obviously diffuses, obviously the inactive form is going down, so we're not at this limit anymore, so you're going forward, and then when you use up enough of your inactive pool that you can't activate anymore, you stop your front. So the place where this is going to stop in this regime is totally characterized by the total amount of proteins that you have. But what's very interesting is we also could see that for the n equals 3 situation, where we didn't have any type of bistability before, we still have this break of symmetry happening. Okay, so this local perturbation analysis, as I showed you, is what we're going to use now from now on to probe these situations. So what's very nice is you can start with a cell where everything is diffusing, and depending where you are, what your parameters are, it can just naturally polarize, or it needs to have a little shove. So in the case of the keratocyte, it needs a shove. Other cells just spontaneously polarize, yeah? So we have these different situations in nature. Okay, but what's very nice about this is that, as Evan also told you, you can talk about this effective surface tension that a front and the back make with one another, right? This isocline. So these are the colors of high amounts and low amounts. Yeah? And it has a tendency to straighten out. That means that if I were to perturb this interface, it straightens out. And we can change these parameters to have the straightening out happen a little bit more quickly. So just bear in mind this effect, because it's going to come later. More recently, together with Alan Champneys, we could take that same system and show that not only do we get the wave pinning situation, which is the one where we have a total amount conserved, but you might have systems where you actually are producing that protein, you are changing those total amounts, and you can get obviously different type of patterns through the very same biochemical interactions, like Turing instabilities, a scalar Maxwell front, and this other thing for which we have now empirical evidence, it's called the homoclinic staking. So basically we have envelopes, and in those envelopes we can have little spikes, right? So we have something very different. Okay. So what am I telling you? I'm telling you that there are mechanisms now, not just to get a front back, but to get like multiple fronts, or to get fronts which are oscillating. And you know, you have a rich set of cell polarity behavior, which can be explained from generic, going to the membrane, coming back in, and having some type of interaction. So then with a student, Katie Abley, we started to think, okay, there's so many stuff out there. What do you think would be like the minimum number of interactions that you would need to get a front and a back biologically? So forget the name CDC42 or whatever, just think about like A and B, okay? So if you have two species, one is essentially autoactivating itself, yeah? B is also autoactivating itself, and they cross-inhibit each other, you're going to get them segregating to these different regions, right? You'll get that. And this is something we'd like to say, like, oh, this would be the most simple form of getting a front and a back, to get this type of simple pattern. And then we started to realize, well, maybe this, although the proteins themselves will have changed, maybe that mechanism is something that, you know, because all the our unicellular ancestor would have had something like this, they had to move around. So when you start evolving multicellularity, which happened 24 or 43 times independently, they think, right? You probably would have taken that mechanism and used it forward. So can we see something like this in plants, which is very different than keratocytes, which are things that vertebrates have? So we started looking also then at this leaf here, at leaves, at the cells of those leaves, and we started making movies. So this would be movies that are in my lab. So some cells just like have a front and a back, and they grow like this. They're the ones on top of the veins. But most of them do this here. It's called pavement cells, right? So they start quite isotropic, right? And then they start making lobes and lobes and lobes and lobes and lobes. When they're really mature, they become crazy. They might have like 62 lobes. Like they're really big, and they continue having those pattern formations. Enriched on the lobes it's a certain type of small G protein called ROB2. And there's a different type of small G protein enriched in the indentations. So just for you to know, so if I have a shape like this, yeah, I call this here a lobe, yeah, and this would be the indentation, okay? And so there are better images now, so when the cell is a little bit more like this, it gets these spots, yeah, and it starts growing these things out. Okay? The whole thing is good. So, but there are other cells in the plant which only make one. So for example here, this is another type of ROP. It concentrates in this spot on an epidermal cell of a root, and that then will start elongating. So they make these very long root hairs. Okay. I didn't put the equations. So we took the equations between ROP2 and ROP6, or at least we looked at the biochemistry, wrote down some equations as you would do, and this was the bifurcation diagram that we got. But the funny thing here, remember auxin? So auxin is this phytohormone, which is really important for meristematic activity in plants. It was shown that auxin can activate those small G proteins via the GEFs. It was really interesting, okay? So there's a receptor for that, which initially was questioned, and now it's been shown again. So we use auxin as the bifurcation parameter, because we know auxin will be something which will be varying during the development. And here we have one of those species, the ROP2, which is the one which likes to locate to the lobe. And so the thicker line here shows what's happening with your global equilibrium, right? And then when we introduce this idea, okay, that we have the separation of timescales, that we have the local, a local patch and a global patch, which are separated, and we have the cytosol, which is diffusing very quickly, we get such a map here. So we see that there's a regime where nothing would polarize, where it might be polarizable if you give a big kick, yeah, then you go to the local lobe. And over here, actually, you go through a turning bifurcation, and you will get a selection of modes. And we can calculate even those frequencies. So to make sure that this is really happening, we look at the simulations, because there we can actually put realistic diffusion parameters. We don't have to take these extreme. And so we see here we don't get a polarity. In this regime here, either we have a little bit, like you think it's starting up, but we didn't give enough, a big enough stimulus, so it's an initial small perturbation. But with a larger perturbation, ta-da, we get a polarity. So where ROP6 is high, ROP2 will be low, and where ROP2 is high, ROP6 will be low. Right? So you have to read them. So this is like the perimeter of the cell, and this is the time. And what we see here is then, as we increase auxin, we get initially more and more of these lobes, and then suddenly we get less of them again. And that was pretty cool, because the experiment showed that if you increase the amount of auxin, you get more and more lobes, and then you get less. So we could sort of quantify that a little bit. So what is actually happening here in the end of the day, though? Because these are simulations where, we're predicting things, we're doing it in 1D, so we wanted to do something now. Let's take a look how this looks in 2D. So we have those ROP2, ROP6 interactions on a circular cell. And this is a cell that we just sort of read in from a confocal image. Okay. And what you see is, you see these ROP2 fronts, ROP2 domains starting to emerge. At a certain moment, I'm going to speed it up. So the biochemistry is the same. There's nothing different between these simulations, except that here we have curvature. We have a shape. Yeah. And these cells are pancakes. So we simulate a top membrane, lower membrane, and the cytosol. And what we see is the shape interacts with the internal biochemistry, with that pattern, because basically, if you think about these isoclines here, right, so which is showing you that frontier between the high amounts and the low amounts, yeah? So this is a certain... because of the no-flux boundary conditions, they want to have a 90-degree angle with that surface, and you start getting distortions due to that in the bend of those isoclines. And because there is this effective surface tension, they tend to get trapped in these lobes. So we have, although if you had a round cell with longer time scales, you would actually go back to a front and a back, because that is sort of the lowest configurational energy, so to say, of this pattern. Having a shape already in place, right? Because this is quite a slow process, you actually will get trapped in these sort of beta minimums. So you basically are going to have, it's not that we picked this one, it's actually quite funny how you get these things. Now, obviously, this is not how nature is doing it, because I took a shape which is already distorted, and I allowed a random pattern to come to it. How it actually works is that you have a shape, you get those spots, yeah? You have things growing out, And that just enforces those spots to stay there because they can't move out due to those curvature effects. Okay. But what I'm showing here is already that there is effect of cell shape to the patterning. And so if we were to consider the biophysics of how those shapes grow out and the time scales of those patterns, we might find the real cause of why we're getting the phenotype, because it's an interplay of both of these things. Yeah? Okay, that's just to show this. So, I'm going to jump this. So we're comparing also different models. We have from the pavement cell model, we had that simple AB model. We have models from the CDC42. And we're doing actually PD analysis, but let me just show this one is this is the nicest one. So there's a student, a mathematical student, and one thing he started to look at is what if we take away this conservation of protein? We don't need to have the inactive and the active be equal to total. So this would be the total AT and the BT population. And the interesting thing is, he could analytically show that you would have these different regions. So the region we're in is here E, where you have a high front, or high back, and a high front. Yeah? Do you see this here? Because the green here is the B star, which is the active B, and the A star is the active A. But you see, you could get other things. Well, first of all, the regions here, the white ones is like, don't go there regions if you want to have polarity, because you won't get any pattern. But you can get other interesting things. You could even get one where you have an intermediate pattern in the middle, right? Or this one here where you have a blackout zone in the middle. So there are different things that can come out, yeah? And the only thing we would be changing here is the total amount of protein. So you see how many ways a cell could actually be changing those type of internal patterns. Okay, rest of the story now, I want to move on. Okay, so cells can break symmetry. Plants and animals, they share these similar mechanisms. And just to tell you, cell polarity can be quite complex. But, we're interested in form and multicellular organisms also. And if you look at that, so if you look at tissues, this is like the bristles of a Drosophila. This is the forming leaf that I showed you before when it's really small, just barely saying, I'm a leaf, I'm coming out. You see that there's certain things, there's certain asymmetries in those cells, and you see how they're all aligned to the same direction, right? So that's called tissue polarity. So here we have these little bristles, every cell has a little thingy here to the same side. Here we have the pin proteins, which are the transporters of oxygen, they're all facing the front. And you might also have, and this is the case of Francois' work, for example, all cells moving in a certain direction. That's also a form of coordination. So how is it that you align these polarities? Be it in the way that you manifest a hair, or that you put a transporter, or that you're moving. So how do you make sure that you're stacking them up properly? Because they don't come as Lego pieces, right? They come as these cytoplasmic things. So we took the simplest model, the AB model, and then we thought, okay, let's couple the cells together with this, and let's just start to think about possible rules how one A star on one cell, yeah, could activate, when it's in an interface, another component on the other star. Because these proteins sit on the membrane. Sometimes they make complexes with other ones, but they effectively will be coming into contact. Okay? So the rule that, So first of all, if we don't have any type of interaction, this is a bit, we don't have anything happening, right? So every cell will just spontaneously, so here it's a homogeneous state, they polarize by themselves. I'm sorry, so what's happening here is blue is black, is B, A is, red is A, and here we have the reaction diffusion in the cell and on the perimeter. And we're just labeling the colors with the concentrations, okay? And so these arrows are pointing to the highest concentration of A. So you can see the polarity directions. So yeah, you would just have them randomly pointing. Now what we say is that A star is going to form a binding outside of its membrane to the neighboring cell membrane that it's touching. And if it forms this complex with B star, it will, just a second, how is this one? Yeah, it's going to auto-activate itself more, right? So you have more auto-activation. So a heterotypic binding auto-activates you more. Same thing for this guy here. A star auto-activates you more. Okay? So this is what we call direct cell-cell coupling. So you're going to enhance your auto-activation term if you have a heterotypic binding. Okay, so you still have your break of symmetry, but on top of that, you're going to have this additional thing. So what happens here is that obviously the red, once you get the polarity, but you need to have the polarization in place, so once you have those random arrows, they start aligning because the red with the blue here will give you extra autoactivation. So you rotate your pattern. Now what you see here is like we didn't put any type of gradient on top, so obviously you're going to have these, how do you call them, frustration, right? So you have here going this, forward, here going back. Now you guys might think, hey, you know, these are spins, this is like crystal, we've seen this before. Probably, but what we're saying here is that instead of having an interaction between them knowing where they're pointing, they have to have it very local, yeah? And there might be a way of describing this at a different scale and going back to your physical models. But our idea is just to keep it to the molecular contact, right? Or to that local touch. But then we had a problem because we also know that our cells of plants are very polarized, and that polarity talks to one another, right? So we have these transporters always located to the bottom of those cells, and here always located to the top of those cells. So how do plant cells communicate? They also have those small g proteins, but they have that damn cell wall in between, which is quite big. They can't touch each other. So how might they coordinate, right? Because we need this coordination, for example, to get now the movie works. To get, for example, the reflex system, which gives you these maximums in these stem cells and so forth. So here we devise the following idea. Well, we do know that one of the things which a cell polarizes are the pins, right? The transporters of auxin. And we know that auxin is activating the GEFs. So we thought, okay, so if A star is promoting, for example, the location of a transporter and promotes the excretion of that molecule, and that molecule then, right, is able to immediately through a receptor, right, do something with the GEFs. In this model, if it represses A star, what we see, and we had to do a repress and not an activation in this case, what we see is that we get a coordination of your polarities, more or less. And why is that the case? Because you don't want to point pins towards one another. That actually inhibits one another, so you actually want it to go like this. You could either have it as repressing A or as activating B. Both of those possibilities would be sufficient. Now, this was quite something interesting for us because these cell walls, they're not thick enough for there to be a gradient. So this cell here sees the same amount of auxin as that cell here, yet they point to different extremes. So let me show you here, for example, in this simulation. So this simulation, we just have a piece of tissue, actually from a steel, and we just allow the cells to polarize themselves by the small g protein model. But because they're locating now pins proportionally to the amount of A star, that pin will bring out auxin, which inhibits A. What you see is that you get the alignment of those cells. In this case, it chose upwards. Sometimes it could choose downwards. Now, it'll always choose up or down. And do you guys know why? Can one of the students say? Do you see how the cells are a bit long in a certain direction? And remember how I said there's going to be a separation of those chemicals, A and B? So does it want to separate like this, or does it want to separate like that? Like this, right? Because this will be the smallest interface. So it always picks up a direction with these reaction and diffusion things that is actually going through the minimum diameter, and so the pins will be put either to the top or to the bottom. And what direction it is, I don't know, but then they will coordinate because they have this indirect communication through this diffusible molecule. Now, it was really fun, because people were thinking that one way that you could explain pin polarity is that, well, if you already have an auxin gradient, you can just tell cells, hey, put things up the gradient. But then it was like, okay, but the epidermal cells, they put it down the gradient. And they say, well, that's because cell types are different, right? Yeah. But then it didn't feel satisfactory, right? Because the gradient is a consequence of the cell polarity being aligned. So who's starting what? Now, the cool thing about this model is that because they have the intrinsic ability to polarize, these little, right, indirectly, they will find a direction to go into, naturally. And because the molecule they're using to be able to do that is oxen, they will generate a gradient for free. So they do, in this case, end up pointing up the gradient. And the way to make them point down the gradient is just to change the rule of who does oxen inhibit. Is it A star or B star? So we find that quite exciting from that perspective. So I still have continue my talk. So up till now, we're just re-emphasizing this notion that there are these patterns in unicellular systems which can be used to communicate when you have multicellularity through direct contacts, or with other proteins possibly. But you can also do it when you have a cell wall if you have something diffusible. But now let's make it more exciting, okay? Everything was very stagnant, right? So we have these cells which were boundary conditions only, right? They weren't moving. Let's put some action in here. So are they, can we start looking at the dynamical feedbacks? So for that, you guys know everything now, specialists on the cellular pots model. So we're going to use that to model the cell, right? As you know, experts, so we're going to have adhesion, we're going to have a cortical tension, and we're going to have an area constraint, right? So these are Lagrange multipliers that we put in, and which more or less fairly capture your cell surface mechanics, right? Like, the cell is a cow type of analogy for biophysics, right? And we get here, like for normal cells, we would get just this foam-like behavior. But what if now we start putting in some biophysics, allowing the fact that if you have high RAC in CDC42, which we knew from the beginning of the story is promoting filamentous movement forward, so it's promoting forces forward, and we allow the high levels of Rho to stimulate contraction, what if we enter that information, right, what I told you before, into the CPM? So I'm not going to bore you with all the details, but would love to explain why we do that, how we did this. First of all, we use the hexagonal mesh to do these simulations, because the way ARP23 is branching filamentous actin. And in this model, we have some extra PDEs. We have a partial differential equation looking at the abundance of filamentous actin, where the barbed ends, where are the capping proteins, and so forth. And what we have here is that they will be promoted or not promoted according to your levels of small g proteins. But the physics comes in play at the leading edge. So the more filamentous actin barbed ends, so these ends which are pushing against the membrane, you have at these sites, you will have actually a force, so you'll be able to extend more likely. So you'll have this copying. At the same time, if you have retraction happening and you don't have too many of them, you're able to lose a site, okay? And we were actually able to use some of the biophysical measurements done in Canada with the ratchet model to be able to get more or less good parameters. So now, our initial cell, which was totally round, it spontaneously breaks symmetry, but it starts moving away, yeah? Because here we have effective protrusive forces going forward, where you have this zone, and here you have retraction, okay? And what you also see is that because it likes to sort of, with very long time scales, it starts meandering, but it's quite persistent, right? And we didn't do any tuning. It sort of adopts, it looks like a credicite, right? It looks like a credicite. And that had to do a lot with the way the ARP23 and the distribution of the angles of the filamentous actin inside. Okay. Okay, so yeah, the velocity of movement is the output of all of those interactions that we have inside. But now look what happens here. So this was quite interesting. In one of the simulations, we forgot to put wrap boundary conditions. And we were like, oh, this is so nice. Oh, what's happening? Ah, it's getting squished. Okay, so there. Yeah, loses a piece. So this is the front. So this is where things are extending. Suddenly it merges there. But it's still sort of moving forward. And then it's starting to get two fronts, because this one goes up and this one's going down. Like the poor thing is being contracted, and it didn't resolve it very well. You saw it lost a little part. Yeah. So we thought, oh, this is horribly wrong. Funny enough, some biologists working with T cells, they say, oh, they do that the whole time. If they ram against the wall, they lose a little piece. But still. At that moment, Léa Cachette, she was talking to some biochemists, and they were saying, oh, phosphonocytides. If we mutate the phosphonocytides in these cells, they lose pieces. it's another level here of kinases, so I'm not going to, but we put that level also in. So that would be between the small g protein level, the phosphoinositides, and the cytoskeleton. But look what happens here to the patterns, because I can give you a better explanation than it's just the phosphoinositides. If the phosphoinositides are very strong, as in the case here, F equals 1, you get it trapped. If you don't have it at all, like what we had before, just the small g proteins, you have this thing. And if you have an intermediate strength of interaction with these phosphoinositides, what you see is that this front is just going to pick a random direction. So what happens actually with these phosphoinositides are very complicated. We had all these other equations now in, but in the end of the day, it's going to make your front more peak-like, right? It doesn't want to be broken. So that cell really just wants to have one front. Okay. Now that's really good, because if it rams into something, it's just going to hoover around. You really can't. We tried. Look, we tried to break that cell. We gave it two gradients here in the parameter IC to try to rip it apart. The normal one will rip apart, this will not. Yeah, he wants to have one front. He's going to integrate conflicting information, but he will resolve his one front. Now, the cool thing is that not all cells want to do that, right? Some cells like to wobble around, some like to have different extensions, some like to have them dynamic. So you can see that you have these flavors of dynamics of how it behaves and how it behaves with the shape that you can start understanding now a little bit better. So instead of just saying, oh, we have more proteins, look at how they look, do some analysis, see what regime it is, what pattern formation regime you're in, and start thinking if this makes sense. Okay, so it's a little bit of that. So now this is a CPM cell, which has persistency through the small g protein interactions. And now we put a load of them together. Okay, I just have some different cell types. Here we show, I'm sorry, if someone is epileptic, please don't look at this. Okay, but so what you see here is that these cells start moving to the left, and these to the right, and they form these streams and coordinations. But you guys all know this from, you guys know this from Boyd's and stuff, right? So, you know, if you have active matter, you're going to get this. Yeah. But what's fun here is that the cell polarity, due to the collisions, it forces the cell to reorientate, and because of those reorientations, you start having this collective behavior. Then I take cells, and I start putting strain, I start pushing and pulling the tissue. So this is a CPM, is it doing something? No. This is a CPM simulation where we don't allow for those T1 topological changes, right? Okay. So basically, we have these cells. Okay, I'll tell you what the movie's going to do. So they start deforming because we're putting some strain, we're putting some force, and we're not allowing them to rearrange through T1s. And what will happen? They can't rearrange T1, so they have to stretch, yeah? Their strain is going to be seen in their shape. And because they will start elongated, their rack CDC row diffusion dynamics is going to reorientate them to try to adopt the smallest axis. So, if people tell you that they put force on a tissue and the cells change their polarity because they're measuring forces, you have to say, well, it could be, but it could also be that they're reacting to the shape change. Yeah? And this is a really powerful thing, because until now, if you don't really dive into the biochemistry and these patterns, you would think that obviously a force is being felt directly, but it could just be felt indirectly through the shape change of your cells. Okay, so I have another 10 minutes? 10, 15, yeah? 5? Came 5 minutes late. Okay, so I'll see how I do with this. So now, what if you just, now this is a story, so changing gears a little bit, where we're going to look at a cell polarity experimentally and start realizing that there's some issues there. Okay, just go here. So I'm going to talk about plants now. Whoop, plants. So plants don't only just shove around phytohormones, they also have to take up minerals from the soil, right? And these are the essential minerals and metals for plants. And boron is one of their most important ones, okay? We don't need it, but they do. And they need it because it's basically the construction material. It's what's cross-binding their cell wall components here. So a plant with no boron is a very unhappy plant. It'll die. it becomes happier, it grows better when it has more boron, but then it starts doing something very weird at high boron concentrations because boron starts killing its DNA. It starts damaging its DNA. So we need to be in this optimal. Now, boron is something that if you're in Turkey, you know it's going to be 10,000 times more than if you're in a swampland. So it really varies, right? So plants have to adapt. They don't know where they're going, they can't walk around, right? They'll have to adapt to the soil that they're on. So they adapt that by, well, first of all, they take up boron by having transporters. On the outside, on the outer facet of cells of the roots, they will have a channel called NIP. Okay, so that lets it come in, but they have to pump it effectively up. So if you have a mutant of NIP, teeny, puny little plants, this is how they look like, okay? But they also have to shove the boron into the main part of the root because it's going to be taken up by what we call the xylem. So that's sort of the plumbing system which takes it up to the leaves, to the fruits and everything, like we eat, for example, right? So we need this. And this is located to inward facing, to the inward tissues, okay, of all of those cells. Okay. So you have NIP on the outside, BOR1, BOR2 to the inside, and they're very polar. Okay, so here we have mutant of BOR1, also not happy, can't get enough boron. And then what we see is that these transporters, and this is, we did a postdoc did, Sotte, very talented. So he started to look at, okay, so this is the transport, the importer. This is its location on the membrane when it has a little bit of boron, so it really wants to be highly expressed because it's letting the boron in. But when boron becomes a little bit too concentrated, it turns it off. It's sort of closing its doors. And the same thing similarly happens with the exporter of boron, which is shoving it into the system. It turns off a little bit afterwards. Okay, that's how regulation happens. That's how they control the amount they get. But what we didn't understand is that if you do this treatment, taking it from low to high boron, so it had a lot of transporters very quickly, and this is borate, very quickly it is taken away from the membrane. And afterwards, when we started to look more closely, like in 20, 50 minutes, it's off, right? It's actually very, very fast. And the same thing was happening with NIP. So I told you that it needs these transporters because it's growing in a soil which could have different concentrations. But why would it need to respond so quickly to a change in boron? Because these boron levels, they don't change significantly in one location. It doesn't really get washed away with water. It's not like nitrogen, like suddenly there's a lot of nitrogen, it needs to go for it. So it didn't make any sense. So in biology, sometimes we can ask this question, why the rush? So why are we doing something, so much regulation on a fast time scale, because it takes a lot of energy, if you're not responding to anything quickly? Okay, so the first thing we did is we made a model, just like this is the medium. This would be your donor cell, the cell where you're giving your boron to it, your xylem. We have your NIPs on the outside and the exporters on the inside of those cell walls, right? And in between we have the cell wall. And so then we just very briefly, just write the transport equations and how those concentrations in the cells, C1, C2, will vary, okay? So the only thing we're taking into account here is that boron is going through, there's going to be a concentration of boron, C1, C2, concentration of boron in the cell walls, and it's going to be regulated here by how much NIP1, N1, B1 you have, or N2, B2, and so forth. But these transporters, they have a regulation that we know. This one is on the transcriptional level, so it's being downregulated by boron. And this one here, its degradation is being upregulated by boron. So this is something which we knew from experiments. So we put that regulation here. And notice here that I put an epsilon in front of that regulatory expression. That epsilon means that when epsilon is 1, I have what we call my wild-type parameters. And when we put the epsilon lower, we will lower the response time, just to see what happens, okay? Because our question is like, why do you respond so quickly? So yeah, so we do a lot of homework. You know, we're trying to get all of these parameters as good as we can, and some we just, guess. And when we look at the throughput in the system, if we have a regulation, what we see is that as we put boron concentrations higher, your maximum amount of boron is not going to go too wildly up, which is good because it gives you DNA damage. Without regulation, you wouldn't be able to exist very far this way. And furthermore, if you look at the throughput of boron through the system at very low concentrations, you do much better with regulation. So obviously, regulation is better. So let's now look temporally. So this is what's happening for epsilon 1. Cell 1 is here, cell 2, cell 3, cell 4, right? So these are the cells all the way up to the xylem. So they have these stable amounts of boron. Now, I put that value lower, 0.1. So now we have 1/10 of the quick response as a wild type. And what we see is we start getting these oscillations. Now, look at this. So I have a coupled system of transporters. I have a medium, which is just a parameter. There's nothing varying in the medium. And you get these oscillations happening. So we could do a stability analysis on why this is happening, but the thing is, what we realize, I'm going to make a big, long story short, is that if you have polarity and you don't respond quickly to little variations, quickly, you will have a traffic jam, okay? So it's the very same thing as, so this is the experiment that they did in Tokyo. Not my collaborator, but a friend of his, and not because of this work. He just happens to work on jams. So, as you know, like all these cars are asked to drive with the same velocity, but if you put the density too high, you will inevitably unstabilize the system, and you're going to get clusters of cells. So what we saw, even if we would put our cells, which we usually think as a medium going to a xylem, in a circle, they also give you traffic jams going backwards. And those traffic jams can be avoided if your reaction time to your response time to your boron and your transport is very quick. So, what I tell the biologists is, you're not responding quickly to the environment or to any different thing you're sensing. You're responding quickly because you happen to be in a polarized tissue. And that puts constraints on how you interact with one another. Right? So to keep things not changing, you have to, behind the scenes, be running around really, really, really quickly. So this is something we're still thinking about how to test it empirically, because it's really difficult. And that was it. So that's a little bit about cell polarity, tissue polarity, and the constraints that tissue polarity gives you, once you have it. So thank you and questions.
Thanks a lot. So, open for questions. We have a couple. Let's start here.
So, from your movies, everything is in 2 dimensions. But what about the 3rd dimension? I mean, why can't you have polarity in the vertical direction also?
Yeah. So, very good. It's quasi 2D, some of them, because what we have is like we imagine that we have a layer of membrane, and then we have a cytosol, and then we have membrane, right? And so we have these compartments, and we're looking at both of them from the top. So that's the first thing. So we do have these rates being different. But we do have actually now a model where we have a 3D cell, right? So you can have these patterns in 3D.
In real life, it's 3D, right?
Yes, it is, but let me tell you. So the pavement cells, they are really long, and they're very, very thin.
Okay.
Yeah, they're very thin. So basically, they're like maybe 1 micron high, 1-2 microns high, and they can come up to 500 microns in length, right? So they're like very flat pancakes, but definitely they have an in and an out.
But they could be vertical, right? They don't have to be horizontal. I mean, doesn't gravity affect them if they're vertical?
Yeah, no, so I was talking about remember when I was showing the leaf? You had those cells that look like this?
Okay.
Yeah, those cells are like, they're like this.
But sorry, so I'm not the biologist, but in real life, are cells always horizontal?
No. They can be block-like.
They can be vertical, right?
Yeah.
But real life, don't you have to play with gravity? That's what I'm asking.
Okay, so you think that gravity would affect what's happening in these cells?
I'm just saying, in your body, the cells can be vertical.
Yeah.
And so the gravity
in fact, in root, they are vertical. Okay, so let me tell you about this. Roots, they only sense gravity in a few cells. So roots are very long structures. They have long cells. It's only at the root cap, where they have starch granules in them, that they can sense gravity. Anywhere else not. And the reason we know that is if we make a mutation here, our roots go up, they go any direction. They can grow in space. It's not a problem. They have absolutely, so this low Reynolds number here is not giving them any sense of gravity per se. Where the gravity is felt is in the root cap. And how that's communicated to the rest of your root structure is through differential flows of a phytohormone that gives them different gradients. So then they can respond to gravity. So if a root is like this, gravity is like this, they will get these differential gradients and move downwards. However, if you do just a couple of changes here, a root would grow curly. It's called agravitropic roots. Yeah? So they do not sense gravity in the cell, per se, but they would have to have, so exactly the mechanics of how this is working is that they think these granules, these starch granules somehow fall and change the cytoskeleton.
Thanks.
If you grow a plant upside down, it can also have the same angle. So it depends where you're sensing gravity. Yeah.
So in your POTS model, what controls the persistence of those migrating cells? So they were highly persistent, right?
Yeah. So, very good. So if we increase the contractility term, the persistency wouldn't change that much, but what would happen, they would start meandering a lot. Yeah. What we think is changing it is the amount of forward push, right? But we weren't able to vary the velocities too much. So it would be nice to look at that.
Yeah, if you include stochasticity in the fields inside the cell, maybe this would reduce
maybe.
the
Maybe it's a good question. Yeah.
Thanks. Yeah, so I have a question regarding the formation of this polarization in the cell. So if I have like a diffusion equation, and I start with a, let's say, symmetrical initial condition, nothing will happen, right? Because I have a diffusion equation, like in every side I will go in the same
No. So if you are in a regime in which your homogeneous state is unstable, you'll polarize it.
Okay, but my question was
just the numerical noise of your simulation.
So it's like a very tiny, let's say,
it's unstable. It's unstable. Yeah.
But that is that the small instability grows up due to the nonlinearity of the term that you put in the reaction diffusion equation, right? So when you show us the way back, I think.
Yeah. Let's go to the. So I think the best way to look at it is if you look at the bifurcation. Sorry. It's very difficult to say. Is it? Let's just go to the simple one. It could be that one or this one here. Sorry. Come on, where are you? This one, yeah? So that spotted line there is unstable. Mm-hmm. So if you're there, I don't know, with IC equals 5, yeah.
Okay.
You start your simulation there, you wait enough, you'll get it coming up.
Yeah, so that term is about, it's the constant that was with the nonlinear term, right?
Yeah.
So, for example, if I, let's say, want to model and try to create a model, and I change that linear term, these results will be the same because you.
Yeah, but look at this system, for example. If I didn't have the inactive form, if I just had the active form, I wouldn't get anything either. You need to have the slow diffusing and the fast diffusing.
But the specific model of the nonlinearity is not so relevant.
It's not. It's not relevant. Okay. I can show you really simple ones can give you things as well.
Thanks.
Yeah, it's a good question. Yeah.
I need to move to the other side.
Thanks, Verônica, for the beautiful talk. I have, I guess, a scientific approach question, and probably a kind of generic question of a physicist to a mathematical biologist. You show us all these models with each time more hypotheses, different ingredients that you add, different parameters, and you even summed it up yourself at the end, which is by saying about the boron model. Well, we have a bunch of parameters that we know, and then some that we just guess. And so this is a big space of models and possibilities that you're exploring.
Yeah.
And
how do you go about that, right?
Well, don't get it wrong, but how do you not get lost, basically? And
yeah. So I would say with the boron model, we were quite happy because the main ones we knew, like the transport, permeabilities, the the abundance of the protein, like lots of things. But there were some things like, what's the rate of breakdown exact, you know, but then you can take a look at similar systems and get an order of magnitude, yeah? That helps a lot. So we have lots of discussions. We never put a parameter in for free, like we're thinking about it, right? However, when we're doing something more exploratory, like, okay, this could be a hypothetical thing, we try to keep it in realistic timescales, but at a certain moment, then you do some analysis and think, oh, this is the interesting behavior, or here dragons exist, right? So you go there and you see what you can, and then afterwards you can think about the mapping. Now also, don't take your model too literally, right? If I have a model which is very generic, you know, those parameters are probably effective parameters anyway, right? So I wouldn't even be able to, you know, to measure them, but I could say what their, so we always try to do something like this. What would be the relative quantities, like the relative amounts? Could we lump them together and say something about who has to be bigger than what, you know, something like that. Like, would the transcription rate have to be much faster than the degradation rate of the protein? So you can start saying qualitative things. But I would say it's an art. So I wouldn't have like one formula, do this and do this and do that. So it all depends on how much you're mapping your model to biology to begin with. Because if you have a one-to-one mapping like we had for the boron model, then you want to be as close as you can. If you already know that you're high, you're like, oh, I'm looking at some Turing patterning, but it's quite abstract. You know, there you can then just make sure that you're looking at all the, or as much of the types of behaviors you can. And this is also why we're working with the mathematicians, because this local perturbation analysis was very, very easy to do because you're taking these limits, infinite, da, da, da. But now they're like, okay, let's say just twice as much, or 3 times as much, or let's say we vary this, right? So they're going through all of that analysis on the PDEs, right? So it's very nice. So there we can, and then they draw up these diagrams of these different behaviors we didn't even know that could exist because we wouldn't have explored it, right? So, yeah. Sorry, there's no answer. You know, we try the best we can, but we do take it seriously if that satisfies you. And maybe with approaches like what you have, where you try to abstract back from a model to get some parameters back, that would be really useful. Like, yeah.
More questions? Okay, if not, we can thanks our speaker
thanks.
again.